Hello, I am Matthew Arsamont, and Jacob and I worked together to produce the experiment Observable Decrease in Motility of Huntington's Disease, Modeled Drosophila and Negative Geotaxis Assay due to Elongated CAG Repeats. Now, what exactly is Huntington's disease? It is a genetic disorder in which instigates the progressive deterioration of the neurons of the brain and is caused by a mutation in the HTT gene, or Huntington gene. Resulting symptoms include dementia, mood swings, uncontrollable movements, which is something specifically that we zeroed in on for this experiment. And the mutation in the HTT gene results in elongated CAG repeats, which is just a portion of the HTT gene itself. Typically, the longer the repeats, the more severe the symptoms of the disease. Then these repeats are translated to form glutamine residues, which interact via hydrogen bonding with other glutamine residues, which form aggregates, and then interrupt the efficiency of the neurons in the brain. Now, a supposed model for Huntington's disease, Drosophila, has many strains that have been made that produce polyglutamine or polyQ repeats. For this specific experiment, we utilized Drosophila with the first exon of HTT gene and polyQ repeat of an expanded length of either 16 or 128 repeat alleles. Once the flies were produced, their mobility was analyzed via negative geotaxis assay, and the effectiveness of the length of the polyQ repeats were deduced, and decided whether the length of the polyQ repeat made a difference on the severity of the symptoms. Now, utilizing the ELAB UAS system, we were able to drive expression of the polyQ repeats so that we can see down here via the Punnett squares that we have the expressive progeny in green and the control flies that we used for this experiment in blue. Once we had these specific flies isolated, we were able to test their mobility via negative geotaxis assay. Despite what this picture down here suggests, we utilized a 50 milliliter graduated cylinder and put a marking about 3/4 of the way up to act as the point in which the flies needed to pass or fail the test. The flies that passed this marker within 30 seconds passed the test, and those that did not failed the test. And this was repeated 5 times for each type of fly. Now, the results can be seen here in the middle with the charts revealing some interesting information. Now, the chart here on the left shows how many flies in total for each type over the 5 trials succeeded quantitatively, like the actual number of flies, and how many failed for each type. Now, it is seen for the males that a much larger fraction of the flies pass the test, and for the females, a much smaller portion of the flies passed the test, and a much larger portion failed compared to the males. Now, I want to explain that males and females, the sex itself has nothing to do with the experiment itself. It's just that the males happen to be the controls and the females happen to be the expressive flies. For the Q128 and Q16 repeats. The sex of the flies have nothing really to do with the experiment itself. Now, here in the middle, we can see the percent success of the flies for each type. And it shows that for the controls, the males, that about 80% for each type were successful. And for the females, it's a little bit different. We had about 58% for both. Characteristically speaking, the males exhibited normal behavior, normal movements during the experiment, whereas the females exhibited less than normal. Specifically, the Q128 had exaggerated jumping and quivering movements during the experiment, whereas the Q16 had a lesser version of that. Now, statistically speaking, we would have expected the Q128 females to have a lower success rate than the Q16, but that is not what we see here. They are equal for the success rate. But characteristically speaking, we received what we expected. The Q128 had exaggerated abnormal movements, whereas the Q16 had that, just lesser, and then the males, the controls, had normal movements. I would like to see this test executed again so that we can get the characteristic results to match the statistic results, And if this were to be done, then I would say that one could look to do more complex tests so that we could directly analyze the brain and then see specifically what pathways are being interrupted by the aggregates forming from the repeats. Thank you, and my acknowledgments and references are displayed below. Thank you.